Hello everyone, my name is Shashi and I will be walking you through the 7A lab today. In this lab, we will perform the first session of conjugation. Conjugation is a part of the transformation conjugation experiment. And we'll also start a new experiment, genetic regulation, and today we will perform the first session of genetic regulation. The transformation conjugation experiment started on the 5th week. This is a long experiment with several sessions, and you will need to write a lab report including all these steps for this experiment. So you need to keep track of what we do each session and start working on the lab report so that you won't be lost and confused. The whole purpose of this experiment is to determine which gene was defective in the gliding bacteria by complementing the mutated gene and observing Winniwood motility. The mutation is on either glide A or glide B gene, and we will be introducing a good copy or a wild type copy of the glide A and glide B gene on plasmid vectors, and we will try to restore the lost function. Based on which gene restored the motility, we can determine where the initial mutation was. The Flavobacterium johnsoniae are not competent for transformation. That's why we transfer the plasmids with the genes to E. coli so that we can use E. coli as a donor for conjugation. These are the two plasmids. We have PSA11 and PDH223. PSA11 has the glide A gene and PDH223 has glide B gene. Both these plasmids have two antibiotic resistance genes, the ampicillin resistance gene and erythromycin resistance gene. One important fact to remember is that ampicillin resistance is only expressed when the plasmids are in E. coli. The erythromycin resistance is only expressed when the plasmids are in Flavobacterium johnsoniae. This is the flow chart to show the transformation conjugation experiment. So first the transformation was conducted and we transferred the plasmids into E. coli and confirmed by extracting the plasmids and running it on a gel. Now we are at the conjugation step. Conjugation is a method of horizontal gene transfer. Horizontal gene transfer is a process by which genes are transferred from one mature independent organism to another. This is how bacteria obtain genes from other bacteria. This is quite different from transmission of genes from parent to an offspring. The DNA transfer from donor cell to a recipient cell. The transmission requires cell-to-cell contact. Donor cells have special pili known as sex pili. They are required to attach and draw the recipient cell closer. In some cases, the pili forms a hollow tube through which the DNA is transferred. In this experiment, we will use conjugation to transfer the plasmids that we introduced into E. coli into the Flavobacterium johnsoniae mutant. The donor cells are our E. coli with the plasmids, and the recipients are the Flavobacterium johnsoniae mutants. We can recognize a successful plasmid transfer by the presence of antibiotic resistance in Flavobacterium johnsoniae colonies and by the restoration of the gliding motility to the Flavobacterium johnsoniae mutant. In today's experiment, we will set up the conjugations. Now I will briefly explain the procedure. You can watch this procedure in the conjugation session 1 video. First, we have 3 tubes, each containing 1 milliliter of Flavobacterium johnsoniae mutant, the E. coli S17 strain with PSA11 plasmid, and E. coli S17 strain with PDH223 plasmid. This strain of E. coli is competent for conjugation. The 2 E. coli cultures are grown with ampicillin in the media. The Flavobacterium johnsoniae mutant is grown without any antibiotics. We will spin this 1 milliliter of culture at 10,000 rpm for 1 minute to pellet the cells. Then we will discard the supernatant and keep the pellet. The washing step is only for E. coli. We do this step to remove any residual antibiotics. Because the Flurobacterium johnsoniae mutant does not have ampicillin resistance, any residual ampicillin in the E. coli will kill the Flurobacterium when we mix these together. So to wash the E. coli, we will resuspend the E. coli pellets in 1 milliliter of CYE media. CYE is a media that we use to grow Flurobacterium johnsoniae. E. coli can also grow in it. Once we resuspend the E. coli pellets in CYE, we'll spin again 1 minute at 10,000 rpm and then we'll remove the supernatant. Next, we will resuspend both E. coli cell pellets and the Flurobacterium johnsoniae cell pellet in 60 microliters of CYE. Now we have Flurobacterium johnsoniae mutant, E. coli PSA11, and E. coli PDH223, all resuspended in 60 microliters of CYE broth. Now we will be mixing these together. First, we need 4 tubes labeled as FJ, EC, FJ plus EC PSA11, FJ plus EC PDH223. Into the tube that is labeled as FJ, we will add 20 microliters of Flurobacterium johnsoniae mutant. This is one of our negative controls. Into the tube labeled as EC, we will add 20 microliters of E. coli PSA11 and 20 microliters of E. coli PDH223. This is our other negative control. We have 2 negative controls. Into the tube labeled as FJ plus EC-PSA11, we will add 20 microliters of Flurobacterium johnsoniae mutant and 20 microliters of E. coli PSA11. This is one of our conjugation tubes. Into the tube that is labeled as FJ plus EC-PDH223, we will add 20 microliters of Flurobacterium johnsoniae mutant and 20 microliters of E. coli PDH223. This is our second conjugation tube. Then we will mix the content in our tubes by gentle pipetting. We won't be doing any vortexing. Aggressive mixing and vortexing can break the pili. Also, Flurobacterium johnsoniae is sensitive to vortexing. Then we will spot the entire content in our tubes onto CYE agar plates. We won't be spreading or streaking. We will label 4 CYE agar plates with the same set of labels we had in our 4 tubes. Then we will transfer the entire content in the tubes to the corresponding plate. 20 microliters of FJ and 40 microliters of the E. coli mixture, 40 microliters of FJ plus EC-PSA11 and 40 microliters of FJ plus EC-PDH223 mixture. We will place these volumes in the center of the plate. This way, the cells will be in close proximity and will allow maximum contact. Then we will let these pots dry and then incubate at 30 degrees for 48 hours. Now let's move on to the second part of today's lab. The experiment is genetic regulation of the lab operon. Today we will do the first session. There will be a lab report that you need to submit for this experiment. In organisms, proteins including enzymes are synthesized from the genetic code found in the DNA. Some enzymes are regulated at transcriptional level to control cellular activities and to conserve energy. These regulated enzymes can be controlled by regulatory proteins that either activate or repress the gene expression. The regulated enzyme synthesis can be induced or repressed depending on their necessity. Inducible systems have repressor proteins that can block transcription of the gene that synthesizes the enzyme. These repressions can be removed by an inducer molecule. Usually the inducers are the substrate itself or a derivative of the substrate of the enzyme expressed by the inducible system. Lac-operon in E. coli is a classic example of an inducible system. Now we will take a closer look at how the lac-operon works. First of all, an operon is a group of genes that are transcribed at the same time, and they usually control an important biochemical process. There are three structural genes in the lac-operon: LAC-Z, LAC-Y, and LAC-A. The expression is controlled by the lac repressor protein or LAC-I. Here, note how the gene and protein names are written. You need to follow the same format when you write your lab report. Lac-operon has two levels of regulation. In addition to the repressor protein regulation, there is also catabolite repression. In this lecture, I will briefly explain each of these regulations. In the lac-operon, LAC-Z gene expresses the enzyme beta-galactosidase. Beta-galactosidase enzyme hydrolyzes lactose into glucose and galactose. When lactose is available, some of the lactose molecules gets converted to allolactose inside the cells. Allolactose is the inducer that can remove the repression on the lac-operon. Using glucose is the most efficient for cells. When glucose is available, the genes related to hydrolysis of lactose are kept repressed even though lactose is available. Lac-operon is only activated when there is lactose present and glucose is absent. This figure shows the arrangement of the lac-operon. The promoter region is where the RNA polymerase binds. The promoter region overlaps with the operator region. The operator region is where the repressor binds. So when the repressor is bound to the operator, RNA polymerase cannot bind. And downstream from the operator region, we have the three LAC genes: LAC-Z, LAC-Y, and LAC-A. And upstream from the promoter region, there is capsid. I will explain more about the capsid later. The repressor protein is continuously synthesized. In the absence of lactose, it sits on the sequence of DNA just in front of the lac-operon, which is called the operator region. The operator region, as I mentioned before, overlaps with the promoter region. When the suppressor is bound to the operator, RNA polymerase cannot bind to the promoter, so there is no gene expression. When lactose is present, a small amount of lactose is converted to allolactose. Allolactose is the inducer, and it can fit into an active site on the repressor protein called the allosteric site. When the inducer binds to the allosteric site, there is a conformational change in the repressor protein. This changes the 3D structure of the repressor protein, and it can no longer bind to the operator region. The repressor protein falls off, and the RNA polymerase can now reach its promoter region. RNA polymerase cannot stably bind to the promoter by itself. Catabolite activator protein, or CAP, stabilizes the RNA polymerase on the promoter. CAP is controlled by the nucleotide cyclic AMP. Cyclic AMP is needed to bind to the CAP for it to function properly. Cyclic AMP is produced only when glucose is absent. This additional level of regulation is called catabolite repression. A catabolite is a substance that can be broken down to provide energy. Glucose is a catabolite. When glucose is present, the cyclic AMP level is low, and when glucose is absent, cyclic AMP level goes up. When there's both glucose and lactose present, because of the glucose, cyclic AMP is not produced. Because of this, CAP is inactive and it cannot bind to the CAP site to stabilize RNA polymerase. Because of lactose, the LAC-I is no longer binding to the operator and the promoter region is free for RNA polymerase to bind. But without the cap protein, RNA polymerase is unstable and the LAC operon is not fully transcribed. When there's no glucose and lactose is present, cyclic AMP is produced in the absence of glucose. The cyclic AMP then binds to cap and forms cyclic AMP-cap complex. This complex binds to the cap site upstream from the promoter and stabilizes RNA polymerase on the promoter. Now the LAC operon is activated and genes are expressed to metabolize lactose. This table shows when beta-galactosidase is expressed. When there's glucose present and lactose is absent, there is no beta-galactosidase activity. In the presence of both glucose and lactose, there is still no beta-galactosidase activity. In the absence of both sugars, there's no beta-galactosidase activity, and beta-galactosidase is only expressed when there's lactose present and glucose is absent. Now let's turn to the genetic regulation experiment. In this experiment, we will use beta-galactosidase enzyme activity as an indicator of the LAC operon activation. Enzyme activity can be measured by the formation of a product or disappearance of a substrate. In this experiment, we will use the former. We will provide a substrate which will be broken down by the beta-galactosidase enzyme into colored products. We will perform a semi-quantitative assay using the intensity of the color to determine beta-galactosidase activity. We will use our results to determine a wild type and 2 mutants of E. coli and determine their specific mutations. We have 3 strains of E. coli, 012, W575, ML308225. One of these is a wild type and the other 2 have mutations in one of the LAC genes. We have 3 kinds of media. We have glucose broth. In the blue cap tube, we have lactose broth in the red cap tube, and we have glucose-lactose mixture in the silver cap tube. We will inoculate a set of 1 glucose, 1 lactose, and 1 glucose-lactose mixture for each of our E. coli strains. We have ML308225, a loop full inoculated into each of our 3 media. Same for W575 and for 012 strains. These will be incubated at 30 degrees for 48 hours. And on the second session, we will look at the beta-galactosidase expression. Think about how the LAC operon will be expressed in these 3 different kinds of media for the wild type strain. In the next class, we'll finish genetic regulation and perform the second session for conjugations. Thank you for listening.